C3 cycle. There are other pathways also. C4 pathway and CAM pathway are two examples. But these pathways actually complements or supplements C3 cycle, and definitely C3 cycle is the one which synthesizes carbohydrates, but C4 and CAM pathway actually help concentrate carbon dioxide inside leaves, and this is we have to see the role of Rubisco here, because there is a process called photorespiration, which actually reduces the efficiency of photosynthesis. So before we go on to see this process, photorespiration, we have to see the dual role of Rubisco here. We have already discussed it. Rubisco is capable of adding carbon dioxide to RUBP as well as oxygen to RUBP, depending on the availability of these molecules, carbon dioxide and oxygen. Now, when it adds carbon dioxide, it goes on to complete C3 cycle. But when oxygen is in plenty and carbon dioxide is in shortage, what happens is that RUBP gets oxygenated. So that will go on to a wasteful process which is called photorespiration, which actually affects the proper glucose or carbohydrate output. It's a waste of time and energy, and it utilizes Rubisco as well as releases carbon dioxide inside mitochondria. This process is called photorespiration, where Rubisco is oxygenated. So instead of 3-phosphoglycerate, we will get another molecule, that is phosphoglycolate. Phosphoglycolate. So this happens actually during intense heat and a high concentration of oxygen. So, and this actually reduces the efficiency of a normal process of photosynthesis by around 25% also. No, the high heat, because when the atmospheric heat temperature is high, what happens is that the stomata gets closed, so as to conserve water inside the cell, inside the plant cell. What happens is that carbon dioxide do not enter the plant cells, as well as oxygen gets concentrated inside the leaf, because the byproduct of photosynthesis, namely oxygen, cannot be released through stomata. So carbon dioxide is in shortage and oxygen is in good supply, so it undertakes what is called photorespiration. And photorespiration, as you can see here, this is the figure which illustrates photorespiration. RUBP is oxygenated to give phosphoglycolate. This is actually, it gives 3-phosphoglycerate also, a molecule of 3-phosphoglycerate, which is channeled to C3 cycle, but along with that, 2 molecules of RUBP go on to produce phosphoglycolate. And this phosphoglycolate has to be, the carbons, those 2 carbons has to be recovered, and for which phosphoglycolate is converted to glycolate, and it passes or travel through this path to get back to chloroplast as glycerate. So the 2 carbons are saved, but at the same time, what happens is that it undertakes it's a wasteful process. It's a wasteful metabolic process, because it undertakes a long route, as well as it results in expenditure of ATP, as well as carbon dioxide is released inside mitochondria, which is a waste product there. Unlike chloroplast require carbon dioxide, but not mitochondria. So this is photorespiration. So what plants, the strategy adopted by certain plants is that they accumulate or concentrate carbon dioxide through some pathways, and these pathways, one of them is C4 pathway, the other one is CAM pathway. Now, first of all, C4 pathway, or the Hatch and Slack pathway. Before we go on to see C4 pathway in detail, we have to be aware of what a C3 plant anatomy or leaf anatomy is. This is mesophyll cells here, then bundle sheath cells here. So this is the arrangement we can see in C3 leaf. And whereas in C4 plants, the arrangement is a bit different. The mesophyll cells actually encircles bundle sheath cells, and this is called Cran's anatomy. The Cran stands for crown. So this is supposed to be a crown-like arrangement. So Cran's anatomy is characteristic of C4 plants, and in C4 plants, the bundle sheath cells are plenty in Rubisco. So the C4 plants, what it does is that the plant does is that they actually the carbon dioxide received is actually received by a phosphoenol pyruvate in mesophyll cell, PAP, which is a 3-carbon compound, and it gets converted to oxaloacetate, which is a 4-carbon compound. That's why you can guess that this is why the C4 pathway is called C4 pathway, because the first stable product of this pathway is a 4-carbon compound, namely oxaloacetate. So this is oxaloacetate. And oxaloacetate actually gets converted to another 4-carbon compound, that is malate, and then it releases carbon dioxide inside the bundle sheath cells along with pyruvate. This pyruvate gets back to mesophyll cell. So like that it goes. We can see this a little more clearly here. This is the mesophyll cell, and this is the this is the mesophyll cell, and this is the bundle sheath cell. Carbon dioxide gets in. Phosphoenol pyruvate accepts carbon dioxide, one molecule of carbon, that, and it gets converted to oxaloacetate. So oxaloacetate, then malate with the help of malate dehydrogenase. Then malate gets past the membrane of mesophyll cell to reach bundle sheath cells, and bundle sheath cells malate is divided. It is actually it releases carbon dioxide inside bundle sheath cells along with formation of pyruvate. This pyruvate gets back to mesophyll cell, where it undergoes phosphorylation to go to phosphoenol pyruvate, or to recover phosphoenol pyruvate with the help of the particular enzyme pyruvate PI dikinase, which splits ATP to release a molecule of AMP and pyrophosphate. So it's a very unique enzyme there. Now here, this is a carbon dioxide which got released inside bundle sheath cells. So this is how we concentrate carbon dioxide inside bundle sheath cells. And this bundle sheath cell will be having Rubisco in its chloroplast stroma. So bundle sheath is actually rich in chloroplast also. So, and the C3 cycle, we know that the C3 cycle actually take place inside the stroma of chloroplast. And the C3 cycle inside chloroplast, inside the stroma, it undergoes C3 cycle now, C3 cycle, to give rise to carbohydrates. So this is these are the enzymes involved: phosphoenol pyruvate carboxylase, then dehydrogenase, and pyruvate inorganic phosphate dikinase. And this is the it's actually 4-carbon intermediate smuggled into the bundle sheath cells. That's what we see here. And Calvin cycle take place inside bundle sheath cells so as to release carbohydrate, like this. And C4 pathway, it limits photorespiration. How? Bundle sheath cells are away from the surface, so less exposure to oxygen. Phosphoenol pyruvate carboxylase do not have an affinity for oxygen, so it accepts carbon dioxide and gets to oxaloacetate, a 4-carbon compound. Then phosphoenol pyruvate carboxylase allows plant to collect a lot of carbon dioxide and concentrate in the bundle sheath cells where Rubisco is present. So this is it. This is how C4 plants. And C4 plants, there are examples for C4 plants here. Some of them are very familiar to us: maize, sugarcane, etc., sorghum, etc. And around 3% of the total number of plants in the world is C4 plants. So, and this is CAM pathway, or crassulacean acid metabolic pathway. This is undertaken by certain plants like cacti and pineapple. the strategy adopted by these plants is that they accumulate carbon dioxide during nighttime when the stomata is open. In the daytime, stomata is closed, obviously to conserve water, loss of water, and to prevent loss of water. And at night, stomata is open so that carbon dioxide can get into the leaves, and these plants actually convert these carbon dioxide molecules to oxalate state and then to malic acid or malate. So basically this is conserved as organic acids, and during daytime the organic acids are split to release carbon dioxide, which will be channeled to C3 cycle. So this is the CAM pathway strategy of CAM plants. It collects carbon dioxide at night, and during daytime Calvin cycle is done. So they save the precious water also. And the examples are cacti, orchids, and more familiar pineapple is there. And the strategy of concentrating carbon dioxide for C4 plants is they actually separate by space. That is the space of mesophyll cell and bundle sheath cell are different. And in CM pathway, it is the separation is by time. That is the day and night times are chosen differently. Now it's actually basically night versus day. And this is the pictorial representation of C3 pathway, C4 pathway, and CAM pathway. And obviously, the C3 pathway is the one which eventually causes or leads to the synthesis of carbohydrates. And these are inhibitors of photosynthesis. There are certain inhibitors which are used as herbicides, and specifically there are certain herbicides which specifically block electron carriage mechanism in photosystem second. Examples are derivatives of urea, derivatives of triazine, and derivatives of uracil like diuron, atrazine, and bromouracil. Bromacil, they are acting as herbicides and blocking electron transfer to plastoquinone. So the plastoquinone will not be converted to plastoquinol. So that's how they act. And thank you very much.